Welcome back to our YouTube channel, Dairy Pathshala, your one-stop solution to all the subjects related to dairy science and technology. So in today's video, we are going to discuss about the theory of spontaneous generation as well as the theory of biogenesis. So it is a hypothesis given by the scientists of those times that living organisms arise from non-living matter. This theory was also known as abiogenesis. So you have to break down this term into two parts, bio and genesis. Genesis, in the simple terms, meaning originating. Bio means life, but here they use abiogenesis. So what they meant to say that whatever the living organisms are arising, they are getting originated from non-living matter. And for that, they have given an example that if you observe a rotting piece of meat, after some days, when the piece of meat is rotting, you can observe the small maggots. These are the small little insects which grow upon the rotting meat or rotting vegetables. So, by observing these maggots on the rotting meat, they conclude the fact that living organisms arise from the non living matter. And the same theory is termed as spontaneous generation. According to them, a vital force may be in form of God or in any other form, it forms life. So the great scientist of those times, Aristotle, he stated that life is being originated from long living things spontaneously from soil or plants or other unlike animals. For example, we know that snakes or toads or frogs or mice they lives in soil. So according to spontaneous generation, the soil, which is a non-living thing or non-living matter, some vital force gives power to this soil or to produce the living organisms like snakes and mice. Another example they give that the manure or some kind of decaying flesh, they give rise to the living organisms like flies or maggots. And this theory was believed till 18th century. We have discussed in the last video that microbiology as a science was started in 17th century and one whole century we were believing that the living organisms arise from some kind of non-living matter due to a vital force which forms life in these living organisms. So according to the theory of spontaneous generation, if you kept a decaying flesh, you can see these maggots forming groups in and around the piece of decaying flesh. So according to them, some vital force is forcing the life in those pieces of flesh so that it is giving a new form of life in the form of these maggots. At the same time, there is alternative hypothesis going on which is known as theory of biogenesis. According to this theory, the living organisms arise from pre-existing life. So this is exact opposite of what spontaneous generation tell us. What spontaneous generation told that the living organisms arise from some non-living matter due to some vital force. But this theory states that biogenesis, which is meaning that genesis originating from life. Bio means life. So originating from life. So living organism arise from pre-existing life. An Italian scientist or physician, he was the first person who disproved the theory of spontaneous generation. And how he did this? So now please keep your focus because from now onwards we are going to discuss about so many experiments, right? So he tested the common idea which we have discussed in the earlier part of the video, that they have given the example that maggots, a type of insect, they arise via spontaneous generation on rotting meat. So what he did, he placed a piece of meat in three different jars. And what he did, he created three conditions. Okay. So in the first jar, he made he made the jar left open. In the second jar, he corked it tightly. And in the third jar, he covered it with a fine mesh gauze. Right. So what he observed after some days, that maggot only appeared in the open container. So there were three containers, right? So the container which was open, maggots were appear in only that container. And no matter how long he left the jar, there is no development of maggots in the other two jars. So he concludes that maggots developed only in meat that flies could reach to lay eggs on. So what he did and what he concludes that since he left open the jar, flies came and laid their eggs on the meat, and to eat that egg, maggots came, and that is how the life is generated. Not because of some vital force which generated life in those decaying flesh. And that is how he disproved that maggots arise from decaying meat. But many of the scientists of his time that he only disproved spontaneous generation for microorganisms because maggots are microorganisms, right? These are not microorganisms. So maybe in the case of microorganisms, spontaneous generation may be true. Who knows, right? Because these are only trial and error. All the scientists, they are just experimenting with one and another thing. So that is how his experiment goes like. As I told, he took three jars. First jar, he left open. Second jar, he left with a mashed gaze. And third jar, he corked tightly. So in the first jar, the flies came and laid their eggs upon, and because of these eggs, the maggots were attracted and generated. Right. In the second jar, since there is no way to flies to lay their eggs upon the flesh, there is no observation of maggots generating from this piece of flesh. And similarly, in the case of sealed jar, there is no development of maggots. After his theory, an Englishman known as John Needham, he again did a experiment which show that microorganisms arose spontaneously. Now it is very interesting and it is becoming like in debate. So you raise one point and your opposition again give the criticizing that point, giving another reason to disprove you. First, we discussed the theory of spontaneous generation, and by an Italian scientist, it was disproved. But again, an Englishman did an experiment to prove that the living organisms arose from non-living organisms or they arise spontaneously. So what he did that in those times everyone knew, even in nowadays, that boiling kills all the microorganisms. So all were clear about macroorganism. So they have doubt regarding microorganism. So since we know that we whatever we drink or eat, we first boil it, right? Because we need to kill the microorganisms. So were also aware about the fact that by boiling they can kill microorganisms. So what he did, he taken chicken broth and he boiled it. Then he took that boiled chicken broth and put it into a flask and then sealed it and he observed it for few days. So what happened? The microorganisms appeared in the tightly stopped flask, which is completely sealed and is boiled also. So what he concluded that life could also be generated from non-living matter, since chicken broth doesn't have any living thing and it is boiled also. There is no chance of microorganisms being present in them. But again, after observing some days, there were microbes. So how they have generated. So, because of his observation, he concluded the fact that non-living material can generate and give rise to life. But again, now the counter argument. Again, an Italian scientist came. He was the professor named Lazzaro Spallanzani. He was not convinced by this fact. And according to him, he given a condition. Like perhaps microorganisms entered the broth after boiling, but before sealing. And this point seems interesting, right? Just imagine what John Needham did. He took chicken broth. Chicken broth, you consider it as chicken soup, right? It is a liquid. Okay. So he took chicken broth, he boiled it. He killed whatever the microorganism present inside that chicken broth, right? Then he transferred that chicken broth into the jar. And then he sealed that jar. So in the meantime, maybe the chicken broth is contaminated with the microorganism. This is making sense, right? That is what the counter argument given by Spelunky. So what he did to prove himself, before boiling the broth, he put the broth in the flask. He didn't boil it before putting it. First, he poured the broth in the flask, then sealed it so there is no chance of contamination through air, and then he boiled. So now you are cutting the connections from both the possibilities. What are the two possibilities? Either some microorganisms present already present in the broth, one case, which is which were killed by boiling the broth. The second possibility is contaminating via microorganisms present in air. This case was cut by sealing the jar before boiling the broth. Then he observed the cooled broth, and after many days of observation, he observed that no any microbes were appeared in the cooled broth. And he concluded with this fact that the microorganisms which were observed in the John Needham's experiment were entered through the air and not because of some vital force which gave rise to those microorganisms. But still, the critics were not convinced at all, and they again giving counter criticism. It is becoming very interesting. Keep listening. It is a very nice story. So now what comes next as a counter criticism point, that okay, that maybe the microorganisms enter through the air, but as humans, we also require air, right? Because without air, we cannot breathe. So what they tell, that since humans also require air, right? So maybe for spontaneous generation, we also require air. Yes, it is making sense. For them, it was. So they were convinced that there is some vital force which is giving some power to non-living things, but to give the power to non-living things to give rise to a living organism, they require air. Now they're giving some modified spontaneous generation theory. So this is how Needham's experiment looks like. So what he did, he took chicken broth, he boiled the chicken broth to kill all the microorganisms present inside it, and then he sealed the jar. So according to him, now there is no chance of generation of microbes if there is no such thing called as spontaneous generation. But after few days, he observed that there are so many microbes present in the broth. So by this observation, he proved the fact that there is some vital force which is giving the power to this non-living thing, chicken broth, to generate life and produce microorganisms. But the Italian priest Spallanzani states that maybe it is in the meantime that after boiling and before sealing the jar, it is because of the contamination by the air, microorganisms were present in the broth and they developed and reproduced. And then that is what we have seen as the growth of microbes. So to disprove Needham's experiment, what Spallanzani did, he sealed the flask first, right? So in the Needham's experiment, before sealing it, he boiled it. But what Spallanzani did, first he sealed the flask. So he cut the chances of contamination through air. Now the organisms are present only inside the broth. And to kill them, he boiled it. And after boiling it, he observed there is no growth, right? So that is why he proved that the microorganisms were not generated spontaneously. But his peers were not at all convinced. And according to them, they told that the spontaneous generation requires air for generate life in non-living things. So to disprove this fact, another scientist known as Theodor Schwann and Franz Schulze, they did some other experiment. Now try to connect all the experiments together. So in the last experiment, what they told, that maybe spontaneous generation requires air, right? So now what Theodor Schwann did, in the boiled media, he passed the air, right? because according to some scientists, air is required for the generation of microorganisms according to spontaneous generation. But this air is passed through red hot tubes. So he heat sterilized the air. And since the air is heat sterilized, there is no contamination of the boiled media by microorganisms. So there is no growth. So this the theory was disproved. In another case, the scientist Franz Schulze, what he did, again he passed the air in the boiled medium, but this time he did chemical sterilization of the air with the use of some strong acids. Again, there was no growth. So control flask receiving untreated air became cloudy with microorganisms. So they have taken two jars, both of the scientists. In one jar, they pass treated air, either with heat sterilization in the case of Theodor Schwann or with chemical sterilization in the case of French Schulze. So there was no growth of microorganisms in the case of treated air. But in the flask in which untreated air was introduced, it became cloudy because of the growth of microorganisms. The experiments were making sense, right? But again, the counter argument came. These experiments were criticized by stating the fact that the life giving power of air, which was termed as vital force, was destroyed in the experimental flask. What they have given the theory that as humans, if you require air, that means air has some power, or it is only the vital force which is making the non-living things able to generate the living organism. So by this, they criticized these meaningful experiments. To disprove all the theories regarding spontaneous generation, here comes the greatest scientist of his time, Mr. Louis Pasteur, at least in the field of microbiology. He came up with his experiment of swan neck. This is the very important experiment, not only in the exam's point of view, but also to know the basics of microbiology. So, what he did, in place of taking a simple flask, he took a flask which is having a swan neck shaped neck, right? It has a bend which is in the form of a neck of a swan. So, first he sterilized that bend through a flame so that he killed all the microorganisms which were attached to the neck. Next, he boiled the liquid media present in the flask and forced out the air from the flask. Now, he made the end open. Why? To disprove the fact that spontaneous generation requires air. That is what the counter argument, right? The people or the scientists who were supporting the spontaneous generation, they were giving the fact that air is necessary for spontaneous generation. So Mr. Pasteur came and told, okay, let's introduce air. No worry, but I will disprove you all. So now what happened due to the swan neck, when the air entered in the flask, since the organisms are heavier than the particles of the air, they settled down in the band due to the gravity, and hence the broth remained clear after years of holding. And clear means that there was no growth of microorganisms even after the introduction of air continuously. Because of the shape of the neck, the microorganisms keep on getting settled down due to gravity in the neck. However, when the clear broth got contaminated with the microorganisms which were settled in the band of the neck, the microbes were grown in the liquid within the matter of few days. So that is what contamination did in the previous cases. So it was not because of air. So this is the very important experiment which disproves the fact or the theory of spontaneous generation, and he was the first person to prove the theory of biogenesis. So with this, we are going to end this video. Do not forget to like, share and subscribe our channel, and please comment down in the below section If you have any doubt regarding this video or these experiments, we are really looking forward to see your review regarding our videos. Stay tuned for next video. Till then, goodbye.